Welcome grade 12s. Welcome to the topic of genetics. Under the topic of genetics, we need to learn about Mendel, some terminology related to genetics, as well as how to set out and solve problems related to the monohybrid cross. But children, we can't move straight onto the monohybrid cross without first understanding the important terminology, as well as knowing a bit of history about Mendel, what he did, how he did it, and why he did what he did. So very important. We cannot understand how to do a monohybrid cross without first learning about the terminology, right? And we also cannot solve the monohybrid cross without knowing how Mendel set this information out, right? What did he say? We also need to learn about his two laws, the law of segregation as well as the law of dominance, etc. Let's meet Gregor Mendel, the Austrian monk, known as the father of genetics. Why? Because he was the first person to actually investigate and provide acceptable suggestions or acceptable hypotheses of how inheritance is brought about. Right. Now, during his time, Mendel observed different characteristics in the pea plants, right? And each of these characteristics, children, had two forms. Let's look at this table. He looked at the seed shape. Some of the seeds were round, some of the seeds were wrinkled. Pea color, some of the peas had yellow cotyledons, and some of the peas had green cotyledons. Another characteristic he looked at was the flower color. Some of the plants had white flowers, others had purple or violet flowers. He looked at the pod shape and he found that some of the pods were inflated or full, while others were constricted pods. He looked at the size of the stems. Some were tall, some were short. Now let's look at an example of a monohybrid cross. Right. In this example, we see a plant with purple flowers crossed with a plant with white flowers, right? And the result, all offspring turned out purple. And when the first generation were inbred, what did he find? Then he found purple and white flowers were obtained in the ratio 3:1. And finally, when you are working out a genetics problem, this is the format that we need to use. Please understand it and master it. Right, let's look at the format of this diagram in front of us of a genetic cross, right? Now children, before we begin a genetic problem, what do? We start by writing down P1. Or if the problem asks for the second generation, you will write P2, right? Now what does that mean? Well, 1 means first, right? So the 1 there means first, and the P means parent. It stands for parent. So P1 stands for the first parents. From there, we find that we have two headings. We have phenotype and we have genotype. Okay, so in this particular example, what are the phenotypes of these organisms we are working with? Very good. You'll find that one organism is white and the other is black. Now we need to work out the organism's genotype. How do we do that? Well, the problem that you will be working with will tell you whether each of these organisms are homozygous dominant, for example, or are they homozygous recessive, right? And then you will be able to work out the genotypes of the organisms. Now, if the organism is dominant, example here, you'll find that white is dominant over black. So to represent the white dominant phenotype, what do we use? We use capital letters to show its dominance. And for the black phenotype, the one that's going to be overshadowed, dominated, be the recessive phenotype. And in this case, we represent it using a small b or a lowercase b. Now for the recessive, we will use small or lowercase letters to show it that this phenotype is recessive. It's the weaker one. It's overshadowed by the white, for example. Okay, so now you will notice that in this example, white is dominant. Yet, we are using the letter B. And I'm sure you must be wondering, why don't we use W instead? You can if you want to. But imagine using a W, a capital W, with a small w, right? Now sometimes it's going to be confusing to the person that's marking your work, okay? So the marker can actually be confused if your big W is not big and if your small w is not small. So rather not use letters that are very similar when they are in capital or in lowercase. Instead, use letters that are different, and that is why we decided to choose B instead of the W. So using the B, we can see that it's clear. We can see the difference between the capital and the lowercase b as well. Right? So what have we learned? We learned that the capital letters are used to show dominant and lowercase letters are used to show recessive. If the organism is homozygous, children, what happens? You'll find that the two alleles are going to be identical. Homozygous dominant will be represented by two capital B's, right? Homozygous recessive. But now, what's going to happen to these two alleles? We must remember that these alleles are found on the parents. Now, children, when the parents are going to produce offspring by sexual reproduction, right? Before that can take place, what's going to happen? Yes, the sperm and the egg must form, right? And for that to happen, what must take place? Very good. Meiosis has to take place. And when meiosis takes place, what happens? Gametes are formed, and each sperm and egg each get only one of these characteristics or one of these alleles. Why? Because we know that gametes, which are the sperm and the egg cell, have half the number of chromosomes than the adult parent had, right? And so what is meiosis? We know that meiosis is therefore known as the reduction division, right? So once meiosis takes place, the gametes are formed. So to form these gametes, how did we get the gametes? To form the gametes, right? We take each genotype and we separate it, right? So that's in a different sperm, that's in a different sperm, that's in a different egg, and that's in a different egg, right? They have separated, and now we have gametes. When sexual reproduction occurs, children, fertilization will take place. And when fertilization occurs, there is a recombination of the same alleles. Remember, B and B cannot cross because they are both sperms, right? A sperm cannot fertilize another sperm. Sperms must fertilize with an egg. This is not possible. Rather, one sperm crosses with an egg, right? So a big B crosses with a small b, right? And they create possibilities. There's a possibility there, which is B. And there's another possibility, B and B. Let's look at the other sperm. So this big B, one sperm fuses with one egg. That's another possibility with a big B and a small b. And the big B crosses with a small b. And then we get another possibility, big B, small b. But then you see that with lines crossing, it can be so confusing and you can make a lot of mistakes, right? So therefore, scientists have created something that we call a Punnett Square. So instead of using a line diagram like this, right, they use a Punnett Square. Please note that both the line diagram as well as the Punnett Square are allowed in the examination, right? It's just that the Punnett is neater, it shows more clarity, and it's less confusing. Okay, let's understand how we use a Punnett Square, right? Children, step 1, what do? We simply draw a table, right? And inside the table, we must have 3 rows and 3 columns, right? So the first row, you fill in your information. Here's it. We want to show the gametes in this Punnett Square. So here's my heading there. Okay, so how I do it is I look on the right hand side, whatever is on the right, I put on the top of the Punnett Square, right? And whatever's on my left, I put on the side of my Punnett Square. So what did we do? We took the homozygous dominant, the capital B, and we placed it on the left side of the Punnett Square. And the recessive genotype, the small b, we place that on the upper part of the Punnett Square or on the top of the Punnett Square. Right? So now you just pair them together. So capital B is crossed with the small b gives you a capital B, small b. Capital B with a small b gives you capital B, small b. Capital B with a small b gives you capital B, small b. And a capital B with a small b gives you a capital B with a small b. Right? So what have we noticed? We noticed that all the gametes forms there turned out to be heterozygous with one dominant and one recessive gene. So once fertilization takes place, you see the offspring, which is known as the first filial generation, meaning the first set of offspring. But if you are working through a second generation, instead of writing F1, we simply change it to F2, right? And that will represent the second filial generation. Okay, so when we speak about the filial generation, we talk about the genotype again of the offspring as well as the phenotype. In this case, all of the offspring ended up being the genotype being heterozygous. And if we look at the phenotype of the offspring, its physical appearance, all of them are 100% white.